Okay, we are talking about antigen antibody interaction, and we have just discussed about the qualitative type of agglutination or hemagglutination assay. For understanding, we have taken the example of blood group testing, and we have seen how we can utilize this concept. Now the second part what we want to discuss is quantitative approach. We can also use this agglutination or hemagglutination assays for the quantitation approaches to understand the quantity of the antigen antibody complex inside a mixture or inside the sample, right? How can we do this? For this particular part we must understand the concentration of either antibody or antigen, because otherwise we cannot get it. We must know the concentration of either antigen or antibody to understand the corresponding concentration of the rest of the other, because we are having the interaction. Remember, if I draw the interaction, we are having antigen interacted with antibody, and what we get, antigen antibody complex, right? So for understanding or for quantitate a particular measurement, for quantitizing antigen or antibody in a sample, we must know at least one, right? So that's a very basic concept. Now in this case, what we can do, we are having what is called a special type of plate, which is called a microtiter plate. It's called a microtiter plate. Here it is. Okay, let me erase this part. It is called a microtiter plate. Here. Okay. Now in this microtiter plate, it is simply having different wells. Okay, many wells. Here say let's write the concentrations, and say here in this particular part let's write the patient numbers. Okay, suppose we're conducting an experiment with various patients. Now what we want to find out using this type of techniques, why usually we use this technique for, is to get any kind of bacterial infection, if there is any bacterial infection present inside a patient or not, what we can find it. Now if there is a bacteria present or bacterial infection present inside a body of a patient, then what will be the symptom? The body must be presenting the antibodies, they must make the antibodies. So if we take the blood from that particular patient, for that diseased patient, we must have antibodies raised against that particular antigen, right? But we don't know what kind of infection he is having, but definitely his body is having some amount of antibodies generated inside his body to fight against that antigen, right? So this is the concept. So what is unknown is the type of antigen or the type of disease or the type of bacteria or virus which is causing the disease. Mostly here is bacteria, we are talking about antigens. And what is known is that there is antibodies generated inside his blood, his or her blood. So we need to check whether this antibody is interacting with any kind of antigen, because antigen antibody interaction is very specific, I've told you. Now we are utilizing the specificity of antigen and antibody. We are using this specificity of antigen antibody to get the answer, right? Now if this antibody is present in his blood, then we provide them in various type of antigens from various type of disease causing agents, various type of bacteria. We need to check in which type of antigen this antibody is binding with. We need to find it out. If we get this value, we can tell yes, this is the type of infection that is having him, right? So we can get this value. Okay, okay, and this is the type. And also we can also get the understanding that whether the infection is going on, whether there is an infection inside a patient's body or not, by simply taking up his serum, like that. How we, I'm telling you here. So this is called a microtiter plate. So what is called, it is called microtiter plate. Why it's called micro? Titer means there are what is titer exactly, I'll be telling you later, but it's the plate is simply small, having small holes. That's why it's called micro titer plates. Now in this case, let me draw this rest of the part because it will be required. Okay, so let us consider this same concept for 1 2 3 4 5 6 7 8 9 patients. So here's a patient number 1 2 3 4 5 6 7 8 9, and what we'll be having on these columns is simply the titer, the distribution or the dilution of a particular concentration of antibody. Now what we are providing in all these wells here is a concentration of a particular antigen. So we are providing the concentration of not antigen, concentration of antibody actually. Okay, so percentage concentration of antibody. Now the dilution of the antibody. So as we are going in this direction, we are having antibody concentration low. That means we are diluting the antibodies as we are going this right hand side. Now here in the very beginning we are having the concentration 1 by 2, and there we are having 1 by 4, then we are having 1 by 8, then 1 by 16, then 1 by 32, 1 by 64, 1 by 128, 1 by 256, then 1 by 512, and 1 by 1024. So here it is the concentration, you can see. So we are having highest concentration here. As we are going, we are falling the concentration, right? And here it is, this is about the patient number. So let me write it here, P, patient number. Now what we'll be doing, we'll be just taking the samples from the patient. So we'll be taking the blood from the patient and simply putting it into these different wells. And what is our concept is that corresponding region of antibodies will be provided to those samples. So if you look for rest of the part, just rub and just look for this first patient. So what we'll be doing, we'll be just putting blood in these places, and what we'll be putting along with this concentration of antibodies. Okay, so what we'll be providing the blood for the patient 1. We are having agglutination, concentration slight less, agglutination, here it is also agglutination, agglutination, no agglutination, and from this concentration there is no agglutination anymore. Okay, for the next one, let's say here it is a agglutination, here it is another agglutination, here it is another agglutination, then no agglutination. Patient 3, for example, here it is, here it is, so lot of agglutination for patient 3, rest of the part is never. Patient 4, here it is, only 2. Patient 5 here, let's say in this case, say here, like that. Okay, like that, say. Okay, so let's say here it is the microtiter plate, and what we get is a result. And what we are doing, we're simply putting blood in this all wells, and not only blood, along with blood we are putting the antibodies. Now suppose this is the well, here we are putting the same amount or same concentration of the blood sample. So concentration of blood sample is same. The amount of blood sample, the volume, not amount, volume, whatever, the volume of blood sample is same in all this, and for all these patients, for everywhere. We're just putting it here. Okay, and what we are putting, the corresponding concentration of antibody. Now in this first well we are providing the amount of blood, then what we provide, this antibody. And obviously what we are making here, we are making two things constant. One is the concentration of antibody, and obviously the volume of blood, volume of sample. We are providing both these things in equal amounts for all the patients. Okay, so here it is in this case, suppose we are providing the concentration of antibodies a particular type, 10 to the power 2. Concentration means simply it's a what we can say, gram per liter, not gram, nanogram. So 10 to the power 2 nanogram per liter, for example, for this case of first one. So this is the concentration which will be constant for all this case. So in the first one we provide the concentration of 10 to the power 2 nanogram per liter onto this, but we take this sample from the half diluted stock. Now for the second we take we provide the same amount of concentration, we take it from 1 to the power 4th. So we are providing all these antibodies in this corresponding concentration, corresponding dilutions in the different wells, and we get these results. Okay, now what are these results is telling us is that as we are going right hand side, we are having less and less antibodies. As we are having in the first, we are having higher antibodies. So that means if there is higher antibody, there is a chance or better chance of agglutination, and these red things are indicating agglutination. So let me tell you, these red things are indicating agglutination. Okay, it is indicating agglutination. Now what we are getting from here is that as we are going right hand side, we are having less and less amount of antibody, so less agglutination. So that's we are getting maximum agglutination at the beginning. So agglutinate, 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 agglutinate. Then from this dilution, 1 to the power 1 by 32, from this dilution there is no agglutination seen for this patient 1. That means this is the concentration, which is the maximum diluted concentration that is giving him the last agglutination, and we are interested about this particular concept. We want to find the maximum diluted concentration of the serum that is providing them the agglutination. Okay, so the maximum concentration, maximum diluted concentration, that is, this is the maximum diluted concentration that is providing us the agglutination, is called a titer. Remember, I've told you it is called the microtiter plate because we are trying to find the titer. The titer means the maximum dilution that is providing us the agglutination. So here it is for the second one, the maximum dilution giving the last agglutination is 1 by 8. For the third patient it is 1 by 64. So let me mark the titers for you. So the titer for the first patient is this one, second patient this one, third this one, fourth this one, fifth this one, sixth this, seventh this, eighth this, and this is for 8, and this is 9th. So these circulated regions are the titer concentration for each of the patients. So if you look for the titer is 1 by 16, here it is 1 by 8, here it is 1 by 32, here it is 1 by 4, here it is 1 by 64, here it is 1 by 32, here it is 1 by 8, here it is 1 by 2, it is 1 by 32. Here are the titer concentrations, right? Now what is this actually telling us? Now for example, let's take this as this patient 8. Let's take this patient 8 as an example. The patient 8 is having only one agglutination when the concentration is pretty high, but just below the concentration they haven't got any kind of agglutination. That means this patient is having a very less, very less amount of antigen in its sample. Very less antigen in its sample, right? Because if it is having higher antigen in its blood sample, then it must have higher amount of agglutination in higher and lower concentration of that, right? So because remember, antigen and antibody interaction depends upon the concentration of both. So if so whatever we are providing from the patient, so if we're talking about the patients, we are taking blood from the patient, right? And we put those bloods there, so we take the antigens from the patient. So the source of antigen here is from the patient, but the source of antibody is simply provided by us. This is completely neutral, we provide this, not patient. So here the termination or the agglutination at the very beginning, high concentration, that means we are having less amount of antigen. If there are a lot of antigens are present, because we are providing antibody in constant rate, if there is a lot of antigens are present, we must see agglutination in both this in this other concentrations also, but we haven't find it. That means it is having a less amount of antigens present. Now for this for this fifth patient, so patient number 5, what we get the agglutination till very 1 by 128. This is a very diluted sample of antibody, but that diluted sample of antibody still giving us the agglutination. That means we are having a high concentration of antigen present in this blood, right? So by just looking at this table and getting the titer concentration, we can tell that which patient is infected, which patient is not. So there is a level, standard level of particular type of antigen antibody interaction which is prepared earlier. By looking at the table and comparing the titer value for each of the patients, we can tell that which patient is infected, which patient is not. Okay, and also for example, say for a type of say pneumonial infection, or for a type of malarial infection, for example, malarial disease, what we get, the value for the titer for the normal is 1 by 8. So if we get the agglutination up to 1 by 8, it's fine. That means you have a very less amount of antigens present in your blood. It won't harm anything. But if it exceeds 1 by 8 concentration titer, and having giving you the higher titer and higher dilutions, that means you're having high and high amount of, so as you're getting that, so as the titer value is high, that means as the titer value is uh, towards the less dilution, towards the more dilution, that means your sample is infected in higher amounts. So if we look for the values, this value will be, so here if we look for that, if we are going to this direction, the value will be less. So the value for titer, so let me write here the value for titer. Remember what is titer? I've told you, if this value is less, if this value is less, very less, that means you are very much infected. If the value is higher, then you have less infection, right? So it inversely related with the infection, or inversely related with the concentration of antigen. We can also tell these things because the concentration of antigen corresponds to the infection, right? So if you are having value of titer high, that means you are having less amount of antigen. If you are having value of titer low, higher amount of antigen. That's the basic concept. Now for this patient, we are having the concentration, let me write it here. We are having here, we are having a particular, for example, say this one. We are having somewhat like this. Sorry. So just like a slide like this kind of value for this titer. But for this patient, what we are having value of titer is this. So for the comparing this patient number 2 and patient number 4, what we can tell by looking at the titer values, that patient number 4 is having higher titer value than patient number 2. That means the patient number 2 is less infected. It is less infected, but this patient is highly infected, or more infected than this patient number 4. But actually this is not a comparative study, so we cannot compare with between the patients, but what we compare it with a standard for a normal human being who is not having any kind of infection. We just compare the microtiter results with them to look for whether there is any kind of increase in titer or decrease in titer. Now, the titer value, I'm not talking about increase or decrease in titer value, I'm talking about the increase in titer. Increase in titer means we are going at this direction. It is not increase in titer value, because as we are going this direction, titer value decreases. So please don't be confused. So either you take the increase in titer only, or you take increase in titer or decrease in titer value. Okay. So here it is the interaction. Here this is the real thing. The value of titer, as we have given you the values, these are the examples, are inversely proportional to the type of concentration of antigen, that means the type of infection. Okay, so that's why we can see here, by looking at this process, we can actually tell whether there is an infection present or not. Okay, and this is the type of quantitative. But you can ask me a question. Quantitative means we want to find out the exact concentration of antigen, but we are not getting any concentration of antigen from this, right? So that's why it is not exactly called quantitative. I am not pretty informed to call this as quantitative process, because it is not at all quantitative. It is a semi-quantitative process, but it is giving us many informations, as we know, many informations that we can get from this particular titer plates, but it is not totally quantitative. So I recommend you to take this as a semi-quantitative process, but not as a full quantitative process. Okay, so it's giving us many insight of the patient's blood, but not the total. So semi-quantitative process. Okay, so this is but another important thing I forgot to mention here. Here you can, if you look for this patient, then you probably will be again scratching your head. What is happening for this patient? Because it is the most logical thing. If you are getting the agglutination in higher concentration of antibody, it's fine. You should get the agglutination in higher concentration of antibody, and should not get the concentration as we are diluting, because the antibody concentration is very less. So you won't get agglutination in lower concentration of antibody. But what we are here seeing, the concentration or the agglutination is not seen at the higher concentration, but it's seen at the lower concentration. What is this? How can it possible? We must have agglutination in the higher concentration. Why not? The answer, the possible explanation for this kind of results, let me tell you, this kind of results occurs. This kind of results happen many times. Now what is the explanation for that? We know that the interaction of antigen and antibody depends upon the concentration of antigen and antibody. Now in this case, the concentration of antibody is so high that it is not enough sufficient to make any kind of lattice like conformation, because I have told you that if the concentration of antibody is higher than antigen, or the concentration of antigen is higher than antibody, then we won't end up with a waste or a high or strong kind of antigen antibody interaction. We often end up with what is called uh, very weak type of antigen antibody complex. So this is the possible explanation, that in this case, in these two cases, the antibody concentration is so high that it won't provide any kind of lattice like structure. That's why in this case we haven't seen any kind of agglutination. So this is a possible explanation. Now if we get this kind of results in all these cases, we call these regions as the prozone effect. So for this type of regions, when we get the agglutination at the higher concentration of antibody but get the result at the lower concentration of antibody, we call these things as a prozone effect. Prozone effects. Prozone, prozone effect. We call it prozone effect. Okay, so this is also a terminology, you can remember this. So this is the explanation for that. So remember, I have told you that this antigen antibody concentration is very important for the proper reaction, and you have seen if they are having higher concentration of antibody, so higher they cannot interact. Okay, so that's it, and I hope that's helpful. Thank you.